Okay, before I go into chapter 5 and like identify that, I'm going to back up one chapter and go into chapter 4, which is all about functional groups. And just remind you what the different functional groups were. So this list is not 100% like doesn't account for everything, but it accounts for most of them. So I'm going to give you most of these functional groups. My open line here refers to the fact that this nitrogen is going to be connected to a carbon because these are the organic functional groups. This is an amino group. This is going to be our carboxyl group or carboxylic acid group. Again, those two names are referring to the same thing. Notice that the C double bond O is terminal because the H is terminal, as opposed to these two carbons which are non-terminal. This is going to be an aldehyde because it is terminal, as opposed to a ketone, bless you, which is not terminal. Then in addition to that, let's see, there's always the phosphate group we could throw out. Bless you. Double bond, single bond, and then single phosphate group.
Is the bottom one in the phosphate group?
Yeah, it's double bond O. I just accidentally put the line right next to the O, and that was kind of my fault. Hydroxyl group. And I kind of think that's mostly everything. There's the sulfohedrils, which contain sulfur. I don't think those would get you, though. What? Ester. Yeah, I mean, typically they don't go over, like, esters versus ethers, so I kind of just skip them. But you are right. Those two were virtually always included, just not in bio. But, one last thing to mention is that both of these, the C double bond O's, as you see them there, they're collectively called carbonyls. But they're just two different types of carbonyls. Cool. So I just kind of wanted to go through that because that's something you definitely will see on the test Friday. You'll see it on the test in real life in May. And then let's go into chapter 5. Which are your biochemicals. And if you're looking to start maybe rereading or looking at again some of these chapters, I really wouldn't focus on 4 any more than what you see in this listing right there. Chapter 5, I would say certainly maybe consider rereading it. One of the most important chapters. Certainly there's the cliff notes on it on the website. And let's see, what are your 5 major classes of biomolecules? What's one? Lipids. And then another one would be what? Okay. Then what would be another one after that? I'd say skip a couple lines in between these. Perfect. And the last one. Yay. So let's talk about lipids. What about them? I mean, just start shooting facts my direction. Do it. Glycerol head. Almost in every case, the fatty acids would be in that category. A modified glycerol head would be in your phospholipids. I think maybe we want to break this down just into like a couple different groups. So you have your fats or your fatty acids. Then you also have your phospholipids. And then what else do you have aside from your phospholipids? Yes, you may. What else? Phospholipids, what else? Steroids. Booyah, steroids. And I would say the glycerol head's going to be present in fats for sure. Because if you remember like what a fat looks like, fat's going to look something like this. And then long carbon chains, hydrocarbon chains, that's what my squiggly lines here are. Cool beans.
And then what's the saturated and unsaturated? What is the double bond for?
The term unsaturated means that you have at least a double bond. So I'm going to write double bond with a possible S. Whereas saturated means that you have only single bonds.
Unsaturated for the liquid type.
Let's see. I have to think about this real quick. Saturated fats, since unsaturated fats have a little bit more kink in them, they'll be a little bit more fluid based, so they couldn't as highly packed. So I always, what's going on in my mind is I think of an unsaturated fat, just to tell you about the process because I don't have that random fact memorized. Is I think of an unsaturated fat. They look a lot like this. There's like two pieces connected to your glycerol head. It's like two. Do you notice how like it kinks upward? That kink is because of the presence of a double bond right there. That's a double bond, which makes it unsaturated. That means if you think about like putting these in stacks next to each other, it's unlikely they would stack very closely because of the kinks. And so that means that they wouldn't be as like solid or stackable. So the unsaturated fats would be more fluid based. Saturated fats would be like they would able to be more like solidified. Cool. All about the kinks in that. Your phospholipids look about the same, except you'll notice that on the phospholipid, it looks something like that. That's what a phospholipid would look like. Remember on the phospholipid, and I always like to draw them like this, and I even orient them tail to tail, which is how you would see them in the cell membrane. Remember, you have a very is that phobic or hydrophilic interior? Hydrophobic, very nonpolar interior, as opposed to a very hydrophilic head. Or I could use the word polar as opposed to nonpolar. Remember, that's how your two phospholipid heads orient themselves. Then steroids, remember they always have a couple of fused rings, and that's the way you can tell that you have a steroid. What's the most common steroid out there? Very good. Cholesterol. Right there. Now the different steroids are going to have different little modifications at these different carbons, and that's what makes them, well, of course, like unique. Cool. Lipids a lot of times are used for a couple of different reasons. I would say like energy storage, of course. What else could we say? Aside from just energy storage. Insulation. Where are phospholipids seen at? Membrane. Yep. What does cholesterol do, or its derivatives like testosterone and estrogen? So communication. Cool. Carbohydrates. These are your sugars. What are your sugars responsible for doing? Obviously providing you energy. What else? Cell recognition, I think, is one of them. Is there anything else that we can think of? What are your different levels of carbohydrates? Y'all can speak up. It's cool. It's monosaccharides, good. What's the next level? Dyes, okay. And then what is the level after that? Yeah, polysaccharides. Y'all remember doing this back in the day, right? Having a good time. Let's see, monosaccharides. Give me the three types of monosaccharides that we should be most familiar with. The names of the three. Good, that's one. Perfect. They all are isomers. C6H12O6. I'll write the word isomer there. Do you know what an isomer is? The same formula but just different structure, which means it has different properties. Like for instance, your body can break down glucose very quickly, but it can't fructose, which is why if you have a lot of fructose intake, then you'll put it on as fat because your body's unlikely to break it down.
Use history.
Why not? Oh, be sure to wear your badge, just in case, you know. Disaccharides. This is when you have what? Yeah, so you have two, right? So give me some disaccharides. Sucrose. Another one. Lactose. Maltose. Good. What's their formula? C12H22L11. You would expect it to be doubled C12H24 though and O12. What's the difference? So that means how do you form a disaccharide from monosaccharide? Is that dehydration or is that hydrolysis? Dehydration. Which means it would be hydrolysis to do the direct reversal. Remember, hydro is water, lysis is to split, so water splitting. That means if you do more dehydrations of more monosaccharides, you'll produce some polysaccharides. And there's 4 different polysaccharides we should be most concerned with. What are those 4, and then where are they used, or what are they used for? Glycogen, good. What else? Good. 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 Hopefully we have all those memorized. Where's chitin found? Without looking at your notes, guys. I want y'all to be... I know what you're doing over there. Like, okay, you got it. Okay, what? You? Insects? That's 1. Where else? Fungus. Exoskeleton and fungus. Cellulose, where's that found at? Perfect. Cell wall of plants. Starch, who makes that? You? Who makes it? Plants. What is it used for? Like, why would they make... Because they're already making the cellulose, so why make the starch? What is that function for them? Can't obviously be for protection, because that's cellulose's job. It's their energy. And I'm going to write down the word human next to it, or I could have just written animal. And I want to write that next to it because I want us to know that you can break down the starches, but you obviously can't the cellulose. That's why they say, like, eat a salad or whatever, because cellulose is insoluble. So your body kind of cleaves it into little pieces, but it can't really absorb it too well. And so it basically, as it falls down with gravity through the digestive tract, it's going to pull off any debris that's like been deposited onto the colon. That's what fiber is, which could do a lot of good things. Most importantly, could prevent like unique cancers, like of the colon. In theory, I would agree at least. If you don't have like a lot of like waste material that's stuck on there, then that would of course be best. Glycogen. What is glycogen? What is it doing? Why have glycogen in this mix of things? Where does it fit into this? Do it? Energy, okay. Energy storage, I would say. And who's doing this, a plant or an animal? An animal. When you go to the gym, it is your glycogen stores that are allowing you to go that short term, very, very, very quick speed. Nucleic acids. 2 major types of nucleic acids. What are the 2 major types? Perfect. And I think you know all about like what's going on with DNA versus RNA when they're used. DNA is the genetic material. RNA is the material that helps to encode or is responsible for producing proteins. It is the middleman between DNA and proteins, the inheritable factor versus the factor that can actually institute change. But But let's talk more specifically maybe about the monomer in this case. What is the monomer of a nucleic acid? What? Nucleotide. And what is the nucleotide made of? There's three things. Okay. 5-carbon sugar, a phosphate group. What else? Phosphate, 5-carbon sugar. Perfect. And then nitrogen base. So when you look at like one of these guys, the picture's typically drawn something like this. There's my B for my base. There's my 1, 2, 3, 4, 5 carbon sugar. And then right here, I have my phosphate group. Now you'll notice that this actually resembles very, very closely to like a molecule of ATP or of GTP. And that's because basically it is. When you have like RNA, RNA is essentially its nucleotide monomer is the ATP that you're familiar with. Now when these line up, when you produce like especially DNA, if we focus on DNA real quick, remember that DNA of course occurs in a double helix, and when you unwind that double helix, the two strands run anti-parallel, do they not? And what is that backbone made of? Sugars and carbons, right? And there's the sugars and then the phosphate groups, correct? And that means that your bases are turned inwards. So, like, if, in other words, if you were to view DNA as basically a ladder, then I would say it's a sugar-phosphate connection that is the side of your ladder. And your rungs, if you will, are going to be made from the different bases. And of course we know that A would bind with T, whereas C would bind with G. What types of bonds are those that are connecting these two bases right here, or right here? Do you remember? Not double bonds, hydrogen bonds. Always good to bring hydrogen bonds back because you remember that hydrogen bonds are like, as a unit they're very, very weak, but collectively they're very, very strong. And typically these bases have either 2 or 3 hydrogen bonds between them. And if you like start accounting for the fact there's like 6 billion bases on one side and 6 billion on the other side, or something along those lines, I mean your DNA is like basically not going to come apart. Cool. Of course, this DNA is packaged along with protein into a structure called chromatin, which is condensed down to chromosomes. And I know we'll talk more about that in the mitosis-meiosis review that we'll do eventually. But yeah, as far as proteins go, how many levels of structure are we talking about here? 4. The primary structure, of course, is going to be the order or the sequence of amino acids. Remember, you create these amino acid bonds or peptide bonds by the removal of water. And the second case, remember, this is when you have the alpha helix or the beta sheet that's produced. The alpha helix or the beta sheet being produced is going to be due to hydrogen bonds being formed. What else do we have? I'm sorry, what? The third level? Right, so this is your R group interactions that are forming. Remember, your R group is different for each amino acid. That's each different amino acid has a different R group, and each different R group, of course, has different chemical properties. So as these R groups interactions happen, it's by this time, once these interactions occur, that you have a full 3D structure. You have what you would call a protein that is conformed to a certain structure. It's like you've finalized the cutting of the key. Basically, you're done. Now, just because Pierce, like, cut me a key for the classroom, is that the only key I would need to operate? Now, there's obviously got to be other keys in the room, correct? There's like other pieces that makes me a whole unit. And that's what the quaternary structure is, which is basically when you have multiple polypeptide units, or multiple peptide units, I should say, that work together as one. Best example of this is going to be HB, which stands for what? Hemoglobin. Hemoglobin is like a tetramer, which means that it's made of 4 different little polypeptide units that all coalesce to form one big unit. 2 alpha hemoglobins and 2 beta hemoglobins. That's the way that works. Cool beans? Cool stuff? Yay. So, I mean, that's basically chapter 5 now. Seriously, I mean, now that means that I'm free to ask you a chapter 5 concept as early as tomorrow on a quiz, if we get to it or if we decide to go that way. There is the great little review, though, on the website. I'm looking at it right now, from the Cliff Notes. And, of course, it's containing more information than this. This is just like the most basic you'd need mentally. But, yeah. Cool. So,